Here's how inflammation leads to type 2 diabetes. It deals with the visceral adipocyte layer. This layer functions in a similar manner that the immune system would in an inflammatory response to cytokines. The visceral adipocytes are fat cells which surround organs and tissues and are very active and very responsive. And not only do these cells generate hormones such as leptin, which functions in regulating metabolism, but they also secrete a variety of pro-inflammatory mediators, including tnf-alpha and IL-6. How this mechanistic trigger works is not exactly understood, but studies suggest that the intracellular stress response produced from the excessive lipid buildup and glucose buildup and fatty acid buildup results in a signaling cascade which enhances the production of cytokines and inflammatory mediators. And this appears to function in a toll-like receptor response because these bind to the cell in question and it binds to the receptors, and these receptors then function in another signaling cascade which leads to the inhibition of the insulin receptor. This signaling cascade produced from here to here and in sequence from here to inhibition of the insulin receptor functions in a similar way that the immune system does in a inflammatory response to recognizing a pathogen. So, as stated, type 2 diabetes is one of the most common chronic inflammatory diseases that we know of. And it's a result of the failure of the cells to respond to the insulin. And this, again, as I said earlier, is a state of insulin resistance. And what does this mean exactly? Well, the pro-inflammatory mediators, which would be this, binds to the receptors, and then the receptors become activated. So this would bind to these, and then the receptor becomes activated. And then once activated, the receptor then goes through a signaling cascade which leads to the activation of this MAP protein known as J and K. And this MAP protein in its activated state is known as MAPK, which would simply mean a MAP kinase, which means that it can now phosphorylate other proteins. So the way the insulin receptor works is the insulin receptor binds to insulin, as you see here. It binds to the insulin. This activates the signaling pathway, and then this pathway then leads to the activation through a signaling cascade of IRS1. IRS1 then becomes in its active state, thus opening the pores for glucose to enter into the cell. And then once these pores are open, glucose can enter into the cell and be taken up and form glycogen branches for storage. This is the normal pathway in a nutshell. Now, what type 2 diabetes does is once the insulin binds to the insulin receptor like so, the insulin receptor then still becomes activated. Once the insulin receptor is activated, it goes through the signaling cascade. But what happens in this signaling cascade is now this J and K protein with its active state is able to phosphorylate, preventing the signal from reaching IRS1. So now the J and K phosphorylates this in the process. This protein is never able to reach its activation state, and as a result, the glucose is not able to enter into the cell. Thus, you maintain high glucose levels. And this is how diabetes 2 type 2 functions, because if you can't complete the signaling cascade, then you can't get glucose into the cell. So no matter how many times insulin binds to this receptor, it still isn't able to complete this pathway. The reason this pathway isn't highly understood is because it's not understood where J and K binds within the signaling cascade to prevent the activation of IRS1. Now, how you overcome this is by oversaturating the cell with the insulin. So there are multiple insulin receptors on the surface of a cell. And what you would basically do is you would continue to bind insulin by increasing insulin production. And when you increase insulin production, all of these receptors are now becoming active. All of these receptors becoming active results in more signaling cascades. More signaling cascades means that eventually these proteins will become saturated by trying to phosphorylate each individual signaling cascade. And once it becomes saturated, it can no longer increase the amount of signaling cascades it is inhibiting. So eventually some of these signaling cascades will be able to make it through to activate IRS1 and thus allow glucose to enter into the cell. The problem is when these receptors become desensitized to insulin because it's overbinding, which is how you reach complications. I hope this helps. If you have any questions, I'll be more than happy to answer them later on today or this weekend. You guys have a great week.